Hello, everyone, and thank you for joining us for this iRepertoire webinar. I'm Alex Parakis with Samba Scientific. Today, we'll be hearing from Dr. Camila Coelho, an instructor at the La Jolla Institute for Immunology. Dr. Coelho has contributed extensively to the field of characterizing B-cell responses to malaria. Today, she'll be presenting her recent work on B-cell sequencing as a tool to improve malaria vaccinology. During the presentation, if you have any questions, we ask that you please submit those using the Q&A button at the bottom of your screen. Please do not use just the chat function, as we will not be able to record your question and answer it during the Q&A session following today's presentation. With that, it is my pleasure to introduce Dr. Coelho. Camila, the stage is yours.
Okay, so good morning, everyone. Thank you so much, Miranda and all the iRepertoire team and Alex for the invitation to speak today. And I am gonna show you some of my work from my postdoctoral training at the NIH. During that time, our efforts were mostly on trying to assess B-cell responses to a transmission blocking vaccine. And I'm gonna tell you what vaccine is that in a bit. And those studies, they were important because we believe that we initiated the B-cell sequencing approach for parasitology in a way that was not so used before. So many B-cell sequencing studies were being performed so far for viral and even bacterial vaccines, but not for parasitic vaccines, including malaria. So that is the most significant part of our study, in my opinion. So just to give you a quick background, Plasmodium falciparum is the parasite that causes malaria. And this parasite has a very, very complex life cycle. And of course, I'm not going over this cycle here with you today, but I just want to tell you that the infected mosquito bites a person, and then this initiates the process within the human host. And usually the field separates or divides malaria cycle in liver stage, blood stage, and mosquito stage. And this nomenclature is also translated to the vaccinology field. So we have liver stage vaccines, which are the pre-erythrocytic ones. We have blood stage vaccines, and we have the mosquito stage vaccines, which are the transmission blocking vaccines. And the most advanced candidates are PFS 25 and PFS 230. And how does this vaccine work then? So in a natural, in a common context, in a malaria endemic area, a mosquito will uptake blood from an infected person, and this mosquito, this now infected mosquito, will carry the parasite to someone else, transmitting the parasite to the next host. However, if this person is immunized with an antibody that targets the parasite inside of the mosquito midgut, when the mosquito bites the person and then bites the next host will not have we will not be receiving the parasite anymore, because these antibodies will kill the parasite inside of the mosquito midgut, and then the transmission will be impaired. And I told you that PFS 230 is one of the most advanced candidates. It is now in phase 3 clinical trial in West Africa. And for this vaccine, we used the domain 1 of PFS 230. And the nice thing about this study is that our lab at the NIH developed the vaccine initiated the clinical trial, and now we had the opportunity to evaluate molecular antibody response to this vaccine. So it was a really translational and important study. And so this clinical trial was performed in collaboration with Dr. Sagara in Mali. And what they saw was that the functional activity of this vaccine, which is measured by transmission reducing activity, meaning that mosquitoes are opened after feeding on serum from vaccinated people, and then we count number of parasites inside of the mosquito midgut. So as less as parasite we have means that the vaccine is being more effective. So we measured the transmission reducing activity using standard membrane feeding assay, which is that mosquito assay I told you. And we found a positive correlation between transmission reduction activity and antibody titers. And you can see that compared to a meningitis compared to vaccine, the activity was really high in serum of vaccines. So we knew that antibodies were playing a role. However, we didn't know which antibodies were those. So what epitopes do they bind to? What is the antibody antigen complex structure? And how do active and non-active antibodies differ between themselves? So we decided to investigate that implementing this platform at the NIH. And for that, I sorted PFS 230 specific single memory B cells. And I shipped those plates to iRepertoire so they could perform this amplification of these 500 base pair fragments corresponding to the VDJ region of both heavy and light chains. And after Illumina sequencing, they sent us back the data, and we were able to perform a clonal expansion analysis to select sequences for clonal expansion. And also iRepertoire also helped us because they have this nice approach of counting repetitions of CDR3s within the same subject. And this approach was really successful in selecting binding sequences. So after selecting those sequences, we expressed them in HEC293 cells and purified those antibodies in column A's, and then we used them for binding and structural assays. And the first thing we saw was that for the heavy chain, we saw that most of the sequences were using IGH V169. So each color here means one subject. And so you can appreciate here that 169 was the most used, followed by IGH V118. For the light chain, we have mostly IGK V4 kappa gene, followed by 1.5, which was found in 3 subjects. And why is this important? Because from those sequences, we end up initially selecting 5 binding antibodies, and 2 of them were very good binders, and they belonged respectively to 69 and 18. And those 2 antibodies, so this is the 69 one, this is the 18 one, they were named after our lab's name, and I named them 01 and 02. So you can see here that using that same assay, feeding mosquitoes, as I mentioned to you, we found that LMIV230-01 was superior in activity compared to 02, although the 2 of them have had a very similar binding rate. So I was curious to see why that is happening. So I decided to check that. And first I did a regular ELISA, and I saw that their binding concentrations was similar in different concentrations. And we performed affinity measurements, and we saw that their affinity was comparable in a nanomolar range, demonstrating that both of them were very high binders, high affinity binders. And I also did this Western blot using protein extract of Plasmodium falciparum gametes. And I saw that both antibodies recognized native proteins in the parasite surface, demonstrating that both antibodies had a conformational exposure to these proteins. So until now, no difference between them. I found the difference when I did this live imaging. So I used live female gametes to stain those parasites with both antibodies. And when I used LMIV230-02, even in higher concentrations, I did not see any binding, suggesting that the epitope was occluded to the second antibody. And I decided to further investigate this functional antibody, which was functional even in lower concentrations. But I was curious to see whether it would bind to other parasite stages in addition to female gametes. And I saw that yes, it bound from gametocytes, which are found in humans, to zygotes, which initiate the development within the mosquito host. And we were very excited about that, but we didn't know about the function of this antibody regarding like how the antibody would kill parasites. And we knew back then that complement activity was necessary for killing parasites in PFS antibodies during infection, but nothing was known for vaccination until now. And we know that there are at least 3 mechanisms that antibodies can kill parasites through NK cells, or through direct lysis, or complement, or phagocytosis. But we knew that during infection, this was complement dependent. So I decided to check that first using serum of vaccines. And I did that with serum, and I found that, yes, there is deposition of membrane attack complex, which are mainly formed by these C5b and 9 protein, which are proteins which are present here in the end of the complement cascade. And these proteins, when they are deposited in the parasite surface, in the pathogen surface, they form a whole, a complex that starts lysing the parasite cell. So we saw exactly that. We saw deposition of those proteins followed by parasite lysis. And I also saw the same thing when I used LMIV230-01, and I really was able to see the holes being formed here, those channels formed here through the deposition of C5b and 9. And I also saw that when I inhibit complement by heating serum at 56 degrees, I depleted parasite development using this antibody, or at least reduced substantially parasite activity. And we also saw that the number of parasites inside of the mosquito midgut in the presence of complement was ablated in the when we had complement, but not when complement was here, when it was not heat inactivated. And since we knew a little bit more about the mechanism of function, we were curious to see whether this was binding in a conserved epitope. So where this antibody was binding to. So we collaborated with Neeraj Tolia's lab at the NIH, and they were able to define the epitope. So here in blue is the epitope, and they used a sequence of 2,500 Plasmodium falciparum parasites deposited in a malaria gene database. And this database provides a sequence from parasites all around the world. And they noticed that those mutations here that are shown in parentheses are the most frequent ones. These 91% and 21% frequent mutations. However, the good news is that those mutations are shown in green. They are outside of the epitope, meaning that inside of the epitope here in black, we have only mutations that were found to be uncommon in those parasites deposited in that database. So we were very excited about that, because we thought that this was a conserved epitope, but we needed to check that. And I got two strains from malaria endemic area. This strain is from Latin America. This strain is from a pregnant woman infected in Mali, West Africa. And we sequenced both strains, and we found that those strains had mutations only outside of the epitope, corroborating what our colleagues have studied, that those mutations were not common. So we decided to mutate those sites here to check whether binding and affinity would be impaired, and we saw that they were not affected, confirming that this antibody has a conserved paratope, and also that it can bind to heterologous strains worldwide. And I was curious to see how common was this antibody among vaccinees. And I developed this competition ELISA assay when I basically incubated LMIV with serum of vaccinees. And I ended up creating this percentage of competition marker, which was based on the frequency of LMIV 230-01-like antibodies in the serum. So as higher was the percentage of competition, higher was the frequency of antibodies similar to LMIV 230-01. And I saw that this distribution was normal, and from 10 to 70% of competition was seen among the subjects. And what was interesting was I found a positive correlation between the prevalence of this antibody serum of vaccinees and total antibody titers in the serum. And most importantly, I found a correlation between functional activity. Here, we didn't use transmission reducing activity. We used a more powerful measurement, which is the transmission blocking activity, meaning that mosquitoes that would have 0 parasites inside of the midgut after feeding serum of people, we would call them transmission blockers, because they would literally eliminate the parasite inside of the mosquito midgut. So we found a positive correlation. However, we saw as well that subjects with low levels of this antibody, but high functional activity, were also in this cohort of vaccinees, suggesting that there is a room for vaccine improvement, or there are other antibodies contributing to the functional activity of this vaccine. And we were pretty excited about this work. We have discovered the first antibody to block malaria transmission in humans. And we were just curious because although this is very promising and can help improve malaria vaccine development, I told you that those antibodies were cloned from B cell receptor. And more importantly, we discussed that this vaccine works by providing antibodies, neutralizing antibodies in the serum of vaccines. So knowing which type of antibody is being circulating in the serum of vaccines, so which antibodies really kill the parasites during vaccination is important. So we were curious to see what was going on in the serum as well. And as you know, both secreted and B cell receptor antibodies, they are identical except for the fact that we have this lipidic structure here to allow that this structure is anchored in the B cell. But basically, we were expecting to see antibody sequences as well. And the other reason why we would be curious to focus in our studies, our efforts in the serum repertoire was because the BCR repertoire is so much more diverse than the serum one. So we cannot use it to infer serum repertoire. So for that reason, I implemented this pipeline in my former supervisor's lab at the NIH. I'm going over through the steps with you. Basically, I just want to tell you first that what is innovative about this study, which was published as a technical advance paper, was the fact that we used antigen-specific single memory B cells and B cell receptors later on as database to match and to identify serum peptides. And we found CDR sequences in both sera and B cell receptor. And this study was the first serum antibody repertoire in response to a malaria vaccine. And for that, we decided to use another set of samples from a PFS 25 clinical trial, which was also performed in Mali. And here we used different type of samples. So here, the plasma samples that I selected, they were either high responders with high titers and high functional activity or low responders. So I had both of them to do some comparisons later on. And I found a correlation between functional activity and antibody titers, demonstrating again that for these vaccines, for this vaccine, antibody is also important. And by the way, I forgot to mention in the beginning, for the other malaria vaccines, antibodies are not as crucial, are not as important for activity as they are for transmission blocking vaccines. So for the other vaccines, we have CD8 playing a major role, gamma delta T cells, and other types of cells. But antibodies are the major functional activity providers here for this type of vaccine. So both 230 and 25 were antibody dependent activity of both vaccines. So going to the platform. So basically what I did was to make a column, a PFS 25 column, to purify only PFS 25 specific antibodies from the serum of vaccines. And after doing that, I used a streptococcus pyogenes enzyme to do a cleavage of the FAB portion, the binding portion, and the FC portion. The FC portion, I froze it. I ended up doing some pilot studies, but I never finished them. But here my focus was on the FAB portion, the binding portion. And we submitted that to liquid chromatography and mass spectrometry. And we used 3 databases to analyze those peptides. First, we used a heavy chain CDR3 database, which was performed using sequencing of total PBMCs. So it was not a so robust approach. But the second one was, we also collected PBMCs from the same subjects to sort PFS25 single B cells. And then iRepertoire again worked with us to sequence those cells. And when we got those sequences back, we were able to have our second database comprised of both heavy and light chain of those cells. And finally, I used IMGT, which is a very well-known immunoglobulin database, to confirm the identity of those sequences. And we used some bioinformatic approaches to define which were the most abundant peptides and which peptides had the highest match between databases. The data is very extensive. We published that, as I told you, as a technical advanced paper, so I'm not gonna tell you about all the data, but I would like to highlight the most important ones. So here, the first data we got was the matching between serum peptides and bulk PBMCs that were sequenced for CDR3, so heavy chain CDR3 from total PBMCs. Here, we got our first hint on V-gene usage and possible association with binding or functional activity. So basically, we found that the most frequent peptides present both in serum and CDR3 heavy chain, they were belonging to IgHV4, here in green. And most importantly, this high containing IgHV4 subject here is the one presenting both highest titer and highest functional activity. So I was very curious about that, and I was curious to see whether there was a correlation between functional activity and V-gene usage. And when Miranda sent us back the data from iRepertoire, we saw that we were correct. So in antigen-specific cells as well, we also had the most increased usage of IgHV4 genes. And again, each color represents one subject. So I decided to match CD4, IgHV4 genes and serum peptides, and also the other genes as well. And what I found was that the highest match, the most identical match, was a IgHV4 gene that was present in both antigen-specific single cell and serum of vaccine. And most interestingly, this region aligned to the CDR3 region, which was very exciting for us. And then I was almost convinced that we had a correlation between V-gene usage and binding or functional activity. So how I tested that? So the story is becoming more interesting now, because after expressing 13 monoclonal antibodies, I found that all IgHV4 antibodies, 100% of them bound to PFS25. However, the other antibodies belonging to other V-gene groups, they didn't bind well. And I decided to test the functional activity of those IgHV4 antibodies, here shown in pink. And I found 2 of them to be highly functional even when I use a very low concentration in mosquito assays. And this antibody here now is being characterized in its epitope, and the manuscript is in final preparation. So we have this very functional antibody, which belongs to IgHVP4. So we are very convinced that there is an association between IgHV4 gene usage and PFS25 vaccine. For PFS 230, we also have our hints, but the paper was not published yet. And we got funding from PATH and Malaria Vaccine Initiative to expand this platform. So we expressed more than 100 antibodies, and we got many binding PFS 230 antibodies here shown in green, and we also got some PFS 25 antibodies. When we did functional assays in a very low concentration, we end up getting 5 very highly functional antibodies, and those antibodies now are being further characterized by my colleagues at the NIH. And with that, I want to thank my former supervisor, Patrick Duffy, which mentored me during this study, all my colleagues at the NIH, and our great partners at iRepertoire, mainly Miranda, which I met through LinkedIn. So I contacted her in 2016 when I was starting my postdoc. And by then, there was no company doing this type of BCR sequencing in so many rare cells. And Miranda decided to make us a pilot. So we ended up being partners in this project very successfully. And thanks to our other collaborators, and I am open to any questions you might have. And I also want to thank my current supervisor here at LGI for his continuous mentorship and support. Thanks so much.
Thank you so much, Camila, for that wonderful talk. So yes, if there's any questions, please send them to the Q&A at the bottom of your screen again, and we'll be sure to answer them in this session now. To get us started, Camila, I guess, first question, what criteria do you use for selecting the binding candidates for expression?
That's a very good question. So this criteria was developed between me and Miranda. So basically, we combined iRepertoire's approach of selecting those repeated CDR3s, plus I constructed some clonal expansion trees using that incantation platform from Stephen Kleinstein. So by selecting the most expanded clones and also selecting the repeated CDR3s, we were able to have a very a very high success rate in binding. So for PFS 230 specifically, this combination of approaches yielded us 97% of success in binding rate. So those were our criteria.
There's another question regarding BCR sequencing. If you have to use mouse tissue, would you recommend isolating B memory cells from the spleen?
That's a very good question. And yes, I would recommend selecting from spleen. We can also use the same approach as we use doing tetramer staining. The only difference is that you need to do an intracellular staining for targeting BCR in mouse spleens. But yes, and I believe also iRepertoire provides this B-cell sequencing for mouse too. Yeah.
Okay, the next question is, Does the transmission blocking vaccine directly protect people?
Yeah, so they do not directly protect people. They indirectly protect people because they impair the transmission. And by doing that, we have a community reduction of parasites, but it's not a direct protection. You help to protect your community. And it is very interesting because I made this study in Mali, asking people how willing they would be in taking this vaccine, even if they were not being protected directly. And 100% of the people said, yes, I am willing to take, because this disease is very severe to those people's lives. Yeah.
Do you see a difference in the properties of antibodies expressed as mAbs versus full antibodies?
That's a good question. I didn't have a lot of experience using the FABs one. FABs ones, we only use 5 antibodies. I didn't see difference in binding, but I've never tested their difference in functional activity. I did test them in spectroscopy for measuring affinity, but not functional activity.
All right, there is another question. Is there a correlation between D gene usage and functional activity in response to this vaccine?
Yeah, so in response to PFS25, as you saw, I believe, yes, there is a correlation. But for PFS230, what we have found so far is that there is a correlation between IGHV169 usage and binding, but not functional activity, which was very interesting. And we are pursuing that in a separate publication.
Another question, is the complement from the mosquito also necessary for the activity, or only human complement?
Yeah, so only human complement is necessary for the antibody activity. We tested complement from the mosquitoes, and they are not required for the activity.
Wonderful. Thank you so much, Camila. This webinar was recorded, so please stay tuned for a recording of this webinar to be posted. And if you do have any other follow-up questions, please send those iRepertoire and we can get them answered offline. Thank you guys so much for joining us for this webinar. We appreciate your attendance and we will see you soon.